Well, this is a project in collaboration among Texas A&M, AgriLife Research, University of Idaho, and USDA that I'm going to talk about today. Uh, First, when we talk about zebra chip, the first thing that comes to mind is potato psyllid, perhaps, and the next thing that comes to mind is browning the tubers. So I'm going to touch on those two topics a little bit real quick. Potato psyllids would they initiate feeding on a plant, inject the plant with the pathogen. If it's early enough, eventually a plant would die by the time of harvest. And if the infestation happens before tuber initiation, then we would have a significant yield reduction and perhaps no tubers. But if the infestation happens a little later and tubers are forming at a time, we may see some browning in the tubers. When you fresh cut them, if you fry them, this brown discoloration would be more pronounced, and that's when the quality loss happens. But there are different variations in the degree of this browning, and we did a study, if you remember, a couple of years ago, looking at how different times of infestation would impact browning in the tubers, and we infested plants with 9 weeks, 8 weeks, until 1 week before harvest with the potato psyllids. And we were able to see that the degree, the symptom severity of a zebra chip, it depends on the time of the infestation out in the field. So the earlier you get the infestation, the more severe symptoms are going to be. But here I'm going to focus on the last two infestation dates, which were conducted 2 weeks and 1 week before harvest. And when we looked at the tubers that were infested 1 week before harvest, visually, if you cut those tubers, they were all negative. And even if you fry those tubers, majority of them, and even maybe 100% of them would fry negative as well. So there was no sign of the disease. And the same if you cut the tubers when plants were infested 2 weeks before harvest, you have a majority of tubers that are asymptomatic. I think in all tubers that we cut, we only had 1 or 2 tubers that showed very faint symptoms. But when you fry them, higher percentage of these tubers may fry positive. So now the question was, okay, does it mean that if the infestation happens late in the season, you're going to be safe, or whether this infestation would remain in tubers and may proceed, or the pathogen would continue to interact with the tubers after harvest as well. So we initiated this very simple study. We harvested tubers. We looked at the percentage of tubers that are tested positive using qPCR out in the field. And then we put those tubers for a couple of months, maybe 2 or 3 months in the storage and 45 degrees, and then we took them out. And when we took them out, it's actually the time zero here. As you can see, during this storage period, we had an increase in the percentage of tubers that tested positive for LSO, and the same were the tubers that were infested 1 week before harvest. There was not much to be detected in terms of the quantity of the pathogen, but when you put these tubers out of the storage and you keep monitoring these tubers, we saw that the percentage of tubers that tested positive increased over time, and the same for the tubers that were infested 1 week before harvest. And remember, these tubers, they all tested negative at harvest. They were all asymptomatic. So they came out of the storage, you put them there, and as we can see, the pathogen actually got into the tuber within that 1 week of time that they had. And similarly, you can see there was an increase in the quantity of the pathogen in both cases when tubers were infested 1 week before harvest and when tubers were infested 2 weeks before harvest. So it made us think that, okay, what happens under a commercial situation? Well, we have the issue out in the field. We try to control it. So if the infestations are late in the season, maybe 1 week before harvest or even 2 weeks before harvest, and the tubers are supposed to go directly for processing, for chipping, then you might be fine and there's not much to worry about. But in many cases, if you're marketing for flies or a lot of tubers that are fresh marketed, they need to be stored and they're being stored for extended periods of time. So in that case, what's going to happen? Are we going to see those effects, continue interaction between LSO and tubers? So we initiated this study that looked at LSO development during the storage time. and we wanted to see if the LSO continues to develop within the storage and whether LSO continues to affect quality of the tubers at different stages of storage. And when I'm talking about commercial storage, it's not as simple as just throwing the tubers out in the cold temperature. There is a process that they go through which involve different steps that I'm going to talk about. And then, based on the results that we find, we would be able to at least not draw a conclusion, but at least to have some suggestions of how we can minimize development of this LSO effect on the tubers, or development of LSO. So what we did was we infested plants with Paetzold 410 and 14 days before harvest. So if you notice, the last time that we looked was 1 week before harvest. Now we narrowed it down. We're going as few days as 4 before harvest. So we infested them with Paetzold. We waited until tubers were ready to harvest. Tubers were harvested, and they were transferred. They were sampled at harvest, and then they were transferred to the Idaho Storage Facility, University of Idaho Storage Facility in Kimberly. And then we continue to continuously sample over time at different stages of storage process. So we harvest, we have a sampling at harvest, then we go through the curing stage where tubers are kept in 55 degrees. The stage could be as long as 3-4 weeks. And the curing stage as well, the tubers have the time to be healed if they're wounded during harvest or whatnot. This curing time would help them to recover from the damage. And then we have a ramp down, which the temperature is being reduced half a degree a day. And Lynn, correct me if I'm wrong on that. And it would take about a week, 3 weeks, maybe a little less, to get from 55 to 45 degrees. And then they're kept in holding temperature, which is at 45 degrees, or that's what we actually did. Again, this temperature depends on the variety that we are dealing with. So we had one sampling before holding temperature, one at the end of holding temperature, and then we let the tubers to sit outside, because under commercial conditions, when you take the tubers out of the storage, they could sit outside for extended periods up to a few weeks, either on store shelves or before being delivered. So what we found was that similar to what our initial study or preliminary data suggested, was that the pathogen continues to interact with the tubers after harvest. What does it mean is that, well, when you had a small percentage of tubers or relatively smaller percentage of tubers that tested positive at harvest, when they go to the end of curing the stage, we had an increase, especially when the tubers were infested 2 weeks before harvest and 10 days before harvest, and the percentages kept going up until after cold storage, after the holding temperature. Similar pattern was observed for the quantity. Quantity of the pathogen harvest was minimal, and then they kept going up. And if you look at these things, the curve, it may suggest that temperature might have something to do with it, but at the same time, you could also conclude that maybe it's just the time after harvest that is affecting this pathogen development, which is something that we need to look at. But again, the distinct pattern is that the tubers that were infested 14 days before harvest, they had a much quicker growth in the pathogen quantity compared to the tubers that are infested 10 days before harvest and the tubers that are infested only 4 days before harvest. So I made this graph to show, one other thing that I want to mention. Again, if you look at the changes when the temperatures are lower, you pretty much see almost no change when the tubers are being kept at the 45 degrees, and pathogens seem to be more active when the tubers are being kept in higher temperature, but it's just observational at this point. So what I did, I looked at the change as well, and the change at the holding period was minimal compared to the rest, or less than the rest. But again, at the curing process, we also had less change, which makes it difficult to conclude that temperature has something to do with it. It might be more of a time. But again, there was a significant difference in the tubers that were infested 14 days before harvest. The change in pathogen quantity was significantly higher than tubers which were infested 10 days and 4 days before harvest. We also looked at some of the quality factors in those tubers. So far, we have looked at 19 different amino acids at the end of curing stage, and then also at the end of the holding stage, which was the 4 to 5 degrees temperature. As you can see in all of these treatments, 4 days, 10 days, and 14 days before harvest, all of them showed an increase from start of the curing, end of the curing, until the holding. So the level of amino acids went up, and I just put these are just 3 examples. Out of the 19 amino acids that we looked at, only 6 of them did not show a significant change in the quantity in all of these treatments. We looked at phenolic compounds, 9 phenolic compounds. Only one of those 9 phenolic compounds that we looked at did not show a significant change, and that was flavanol glycoside. One of those derivatives, I think it was derivative 1, and Chris could provide more information on that. But again, all those phenolics, they showed an increase throughout the storage process. Glucose and fructose, the reducing sugars that we looked at, they also showed a significant effect. But the interesting thing was that, unlike previous studies, that we always showed that the increase in LSO was correlated with an increase in reducing sugars. Here we saw a negative pattern. So actually the increase in LSO was associated with a reduced amount of reducing sugars. And there might be some interactions with the storage process that is going on, and that needs to be looked at in more details. And this was actually the first historical study that we did in this in such detail. And then here I just added data from our LSO in the same graph, so you can see that they correspond to the increase in the LSO quantity as well as the LSO quantity in all of the treatments. It increase between the time of curing until the end of the holding stage. So far, we haven't had the chance to analyze the numerous number of samples that we had, but what we looked at was a percentage positive at harvest, end of curing, and the end of holding. So as you can see, there is a consistent pattern across both years, although there is a difference in the amount that tested positive. Again, like the pattern stays the same, and I'm pretty sure there's going to be no significant interactions there. But the reason that we have much higher LSO in 2013 might have something to do with the stress factors that we had out in the field in terms of drought, and then we had some herbicide drift, field as well. So that might have affected this finding, but still the pattern stays the same. I put this graph to show that I did some stats, but it's not in much details at all. The first 3 on top, they're showing the relationship between amino acids and LSO quantity at the end of curing stage. As you can see, there is not much interaction between these. Again, I just selected examples. There was no significant interaction at that point, but at the end of the holding stage, at the end of 45 degrees, when we pulled out the tuber, significant patterns will start to appear. We have significant patterns in amino acids and phenolic compounds. And this is again, the negative relationship that was not expected, and we need to look into that in more detail. So as I mentioned, out of the 19 amino acids that we looked at, 9 of them showed no relationship with the time of infection. It was not affected by the time of infection. Sucrose concentration was not affected by the time of infection, but we had effects of the 2 reducing sugars that we looked at. One of the phenolic compounds that we looked at was not influenced by the time of infestation, as I mentioned. And then glutamine and sucrose were the only components that were not affected by the time of sampling during the storage at the stage of the storage process that we looked at, all those continuous sampling. So pretty much every other compound showed some variation throughout the storage process, which was not surprising, of course. We also looked at the reflectance measurements of these tubers that were stored, and the effect was not significant, but the negative correlations we expected to see existed there. So we basically, when we took out those tubers out from the storage, from the holding temperatures. We fried them to look how they would fry, and we took reflectance measurements. And the higher the reflectance, it would indicate that the tubers were healthier. So there was a negative relationship between LSO quantity and the reflectance measure we got, which again shows that yes, there's going to be some changes in the quality of the tubers during the storage. So what we found was the LS would continue to interact with the tubers after harvest. And now that we know this, we are always looking into resistance studies and developing resistant varieties and genotypes. And perhaps instead of only looking at the field, we need to continue this observation throughout the storage as well, because that would be an important component to be considered, especially when we have tubers or varieties that need to be stored. Changes in quantity may be affected by temperature, by the stages of the storage that expose tubers to higher temperatures, but we need more studies to pinpoint this factor. It could be just the time. The longer you keep the tubers, the higher chance of finding the tuber or having increase in LSO quantity. But again, the pattern, some of the patterns that we saw in the first year still kind of suggest that higher temperatures increase the interaction between the LSO and the tubers. So the recommendation for now would be, if it's possible, move the tubers into the storage as soon as possible. And there are other factors that could affect interaction between LSO and tuber after wine kill, which Charlie is going to be talking about it later. So some of the practices that we have, post-vine kill practices, let's put it that way, could be manipulated so that we just minimize pathogen development in those tubers. And with that, I'm going to thank funding sources and all people who help with sampling the tubers in Charlie's lab, Nora's lab, and my lab.